This Week in Parasitism is brought to you by the American Society for Microbiology at microbeworld.org/TWiP. This Week in Parasitism, episode number 84, recorded on February 25th, 2015. Hi, everybody. I'm Vincent Racaniello, and with me is Dickson, Bald Eagle Despommier.
Hello there, Vincent.
I say Bald Eagle because I tried to do the intro once, and he interrupted me because he saw a bald eagle out my window.
I did. I mean, I have a
You
view.
did, and you did.
I can see California from
I know.
here.
It's very unusual, right?
Yes. It's extremely unusual to see bald eagles, A, but they're making a huge comeback, and B, this is a very cold day. It's in the middle of the winter, and here is a bald eagle.
All right, hold that thought. Also joining us today, right here in the studio, Daniel Griffin. Welcome.
Oh, it's great to be here.
Do you like eagles?
I love eagles.
Are you a bird guy?
Out in Colorado, where I spent
a lot of time,
20 years of my life,
Right.
We were starting to see the eagles come back. And, as that guy was making, the eagle looks beautiful, but it's scavenge. And the worst is, you see these movies, you hear this wonderful eagle cry. They actually dub in like
That's
a
true.
red-tailed hawk.
This is very
Because
true.
when you actually hear an eagle, it's not impressive.
not a big deal, yeah.
It's kind of like
It's going to screech.
Yeah, it's like a little tiny, not much.
In Vancouver, where they were everywhere in every tree.
Yeah.
Tons of them.
So we're going to genetically engineer eagles that hunt and have a wonderful call.
Maybe we could put a nanobody in them.
Exactly. We're going to do it with camelid nanobodies.
Cross them with minor birds so they can speak.
Yes.
We have quite a few case emails
Great.
today.
That is great because
This is
we only
wonderful.
posted this just a few days ago.
Yeah.
that's
So
very
we have
wonderful.
from episode 83, which is the previous, and from the previous one, 82. And I thought we should read just briefly because
Sure.
I like when people send in.
You bet.
You remember that the 82 case was a malaria in a young man who had gone to Guatemala, right?
Yes, and malaria vivax.
5X,
Indeed.
right?
That's right.
And we may have this noise,
We may.
which is
I
a
didn't do
dentist.
that.
It's a dentist drill.
This
right?
is, that's exactly what it is.
Now with the directional mics, are we gonna
It's
hear
probably not going to be all that prominent. Yeah,
I can feel it more than I can
you
hear
can feel it.
that? So if we're a little off today, it's that we're suffering from this background noise, but hopefully our listeners are not.
Exactly.
But we will. What is the British saying? Keep calm and carry on.
We shall.
Yes.
Stiff upper lip.
Alan writes, my guess is malaria, most likely Vivax or Ovale. So he's pretty
Beautiful.
good.
That's
How
great.
would you know the difference?
Well, two ways. One, if you think about it,
Right.
is there Ovale in Guatemala? So the geography is great. I mean,
Right.
Vivax is really something we're seeing here. Ovale is all over West Africa, and that has the whole Duffy and other
That's right.
things going on.
Mostly
but yeah, the
it's a
geography
West
helps
African
us. Yeah.
distribution, though. That's right.
Alan also writes, on another note, I listened to TWiP while driving home on the Stitcher phone app, which, much like a radio, lets me listen to a list of my favorite podcasts and alerts me when there's an updated episode. The audio podcasts are small enough that I stay under my allotted cell minutes and don't have to download or delete. I actually discovered TWiP via the Stitcher app about 3 years ago, and while I have no financial connection, I have never heard you mention it as a conduit. Keep up the good work. Alan, by the way, is in Hawaii.
Nice.
I do have the Stitcher app, and in fact, they asked me years ago to put my content there because they had gotten requests. So Stitcher is another good conduit. The reason I didn't mention it, Alan, and this has nothing to do with anything except they don't. I can't have their statistics.
You mean you can't access them?
I can, but they're separate from everything
I see,
else,
I
and it's
see,
kind
I
of
see,
a
I
pain.
see, I see.
Although I shouldn't complain about that. But Stitcher's a great app too, so
Aha.
it's fine. Richard writes, my guess is P. vivax malaria. The patient has fever every other day, tertian fever, with shaking, chills, general malaise, and headache, and the labs are all consistent with malaria. This could either be a recurrence with the symptoms of the primary infection masked by mefloquine treatment, or a long incubation period may be due to the thalassemia minor. Either way, a 6-month asymptomatic interval is, I think, not unusual with Vivax. Add primaquine.
Bingo.
Columbus, Ohio. Richie.
See, they know about malaria in Columbus.
Now, Richie wrote again. Hey, guys, I'm loving TWiP 2.0.
Good.
I'm a medical student considering a career in infectious disease.
Oh, great.
And tropical medicine in particular. TWiP was always great, but the added clinical component has improved it. I also love the commitment to more frequent episodes. How come when I told you to do it more, you wouldn't, but when Daniel does, you
Peer pressure.
because he's an MD, that's
It's
why.
all peer pressure.
Regarding the case presentations, I like the structure in 82 with Daniel supplying the basics and Dickson and Vincent asking follow-ups, but is there any way you guys could limit the guessing at that point? I personally think it would be more fun to just present the history and selected other findings and then save the differential diagnosis for the following episode. For example, the discussion of the first and second cases included the correct diagnoses. I don't yet know the answer to the third case, but I was pretty sure it was Vivax, even before Dickson began talking about why that was probably it. I think it would be more satisfying to form a diagnosis from the presentation alone. If I'm wrong and it's not Vivax, well, that undermines my point entirely, and please disregard this email. I think it's a good point.
No,
So,
and
yeah,
I think
that's-
we
that's
started
fair
to do
enough.
that right in the last episode
Yeah,
where I
that's
said,
fair.
you guys think
That's
about what
fair.
you want to think, but don't mention any. And I agree with this.
No, that's good.
Columbus, Ohio, where it's minus 13 Celsius.
We can brainstorm the previous case on this episode.
All right, now this is for the TWiP 83 case,
Right.
which was...
Should I remind everybody?
I
before we
can't
read it?
remember.
Why don't I remind people? This was
I
a
remember
case
now,
of
yes.
a 56-year-old gentleman. Remember, he was from Georgetown, and we
Right.
we initially were bothered till we realized it was Georgetown, Guyana, down in South America,
Exactly.
which is a little country just east of Venezuela. And he was brought in because he wasn't bothered, but his son and his daughter-in-law, who live in Western Long Island, were upset because of the appearance of this man's right lower leg. And he had years of increased swelling of his right leg.
Right.
And there was an open, non-healing wound on the inside surface of the lower leg, just above the ankle bone.
Yeah.
And he felt fine. Wasn't bothering him.
Yep.
So that was our...
So the correct medical term for that is anergic or anergy. He was suffering from anergy because he couldn't sensate. There was no sensation at the surface level of that foot. Would you agree with that or not?
Oh, are we going to read the emails first? Should we read the emails now? Or
what
should
are
we
you
let him
asking?
guess
What are you
more?
asking?
I was just asking to explain further the correct medical description of this lack of pain.
Yeah, I would say it doesn't hurt, but there isn't loss of sensation.
Okay.
And I think
Okay.
that is important.
Oh, okay, fine.
All right, we have a couple of guesses here.
We do.
First one is from Jan, who writes, thanks to Google, I think it's lymphatic filariasis transmitted by the Culex mosquito. Love the Twix shows where I learn a lot outside my normal field, which is trees. We are still in the dark ages there. Our treatment options after diagnosis are rather limited. Improve the soil, let's wait and see, amputation, or euthanasia. Sadly, it's mostly the latter due to economic reasons. Yeah. The weather here in Rotterdam, the Netherlands, is 7 degrees centigrade, and showery wind is not too bad for a change.
By the way, that's the origin of Dutch elm disease. I wanted you to know that it wiped out almost all the American elm trees. So that
came from the Netherlands?
Yeah, this was called Dutch elm disease to begin with. So maybe Jana's an expert on Dutch elm disease. Who knows?
All right, the next one is from Elise. Dear TWiP Trifecta, I'm a longtime fan of yours and think you're terrific both as a duo and a trio. Thank you so much for your work. I am not a scientist at all, but I love listening to you and do my best to follow both TWiP and TWiV out of pure interest. Of course, I've been following along with your recent case studies and have taken notes and done my own non-scientific, non-academic searches to try and diagnose these patients. I was privately pleased that I figured out the babesiosis diagnosis in the gentleman on Long Island by myself, but did not write in due to embarrassment.
You would not have considered that an embarrassing response, believe me.
If you were on the show, maybe.
Even if the answer is wrong, it's not
exactly.
embarrassing.
It's a calculated guess.
Okay, with this new case, I'm feeling reckless, having gotten one right. I think the gentleman from Guyana may have lymphatic filariasis. Here's why. The nematome worms that cause the disease are common in Guyana, and this gentleman didn't drink filtered water, or take other precautions. An infection can take years to manifest while the worms multiply, and many infected people are asymptomatic, so he wouldn't have had cause to complain much, which he hasn't done.
Right.
And what is behavior somewhat typical of many men I know, he still didn't feel he wanted to do much about it, even with significant swelling and an open wound. Dr. Griffin did say that the man's genitals were normal, and while it is apparently common for the genitals to become swollen, perhaps this man is fortunate, since not all have this symptom. Edema described in patients matches the sort that Dr. Griffin described, non-pitting and brawny with skin color and texture changes. In reading about filariasis, I was a bit confused because there is not always a mention of a chronic abscess or wound, but apparently a single abscess can be caused by collections of dead worms on the lymphatic tract, or so that symptom fits. I was interested to see that for diagnosis, blood collection should be done at night because that is the time when the microfilariae are circulating. Does this mean that diagnoses are often missed because blood is most often drawn during the day, or is there so much suspicion about what is causing this constellation of symptoms that medical practitioners know to draw blood at night? Thank you very much for your podcast. If I get this wrong, I hope very much that my having written is not something embarrassing to me. And if I get it right or close to right or wrong in an interesting way, perhaps it is due to the way you explain everything so well and are such good teachers that even someone with no training could play at-home detective. It is very cold here in lower Manhattan, where I am right now, -5 Celsius.
It's not any better in upper Manhattan, by the way, I must tell you.
It is freezing.
Next one is from Ken. I have been listening to the Twix series for a long time and even got to experience the TWiP bump by appearing in an episode of TWiP when Vincent visited MedImmune last year. Having done postdoc work in a trypanosome lab at the University of Buffalo, I particularly enjoy TWiP, and I'm really enjoying the recently revamped format. During episode 83, you were talking about primaquine and a MASH episode in which it was discussed. The character was Klinger, who was a Lebanese American. According to what I found on Wikipedia, not always a reliable source, but I think it is now, hemolysis is an adverse effect of primaquine in Africans or Caucasians of Mediterranean descent. Also, I wanted to take a stab at the case study in episode 83. My diagnosis is lymphatic filariasis due to Wucheria bancrofti. I would recommend doing blood collection at night since the filaria circulate at night in the blood, and then doing a smear for identification. Looking forward to more excellent episodes of TWiP. Hey, Ken, how are you? I remember Ken at MedImmune. And we have one from Kurt. Didn't write in last week because the time course threw me off of malaria, which was my only suspect. The epidemiology time course and some of the syndrome is right for elephantiasis, secondary lymphatic filariasis. However, while lymphatic filariasis may produce numerous bacterial infections, I would anticipate these to be either short-lived or proceed to sepsis. As such, I anticipate that the persistent, unhealing, painless wound would demonstrate leishmania as a mastigotes under tissue sample light microscopy. At this stage in the disease, it's clear that at least some of the words have died, which complicates testing, but he may show some microfilaria in a thick blood smear. If the sample is taken at night and some worms persist, check for splenomegaly and perform an IG assay to rule out visceral leishmania. Refer the patient to a lymphatic specialist for possible elephantiasis therapies. Active filariasis has been known to respond to doxycycline since we're on the cheap, and treat the leishmania with metefosine. Just a couple more here. See, this is great. We had 3 more. Wink, our friend in Atlanta. At 1 hour and 11 minutes, I'm saying Wucheria bancrofti. P.S. The Vivax was at 9 months. Okay. Bill writes, TWiP trifecta. I love the new format with the case studies. Came across Twix Trio a few years back, and they gave my mind a welcome diversion during my commute, where I spend my days managing a team of talented engineers in the auto industry. I was disappointed to see just one response to last week's case, so I thought I would give it a shot. This week sounds like lymphatic filariasis.
Who said the United States public is not well educated? These are non-physicians. These are people with other jobs, and they're all coming up with amazing diagnoses.
I would like a thick blood smear with an H&E stain, collected as late as possible. Should help to confirm the presence of microscopic nematodes. Currently 3 known vectors, Aedes aegypti, Culex, and Anopheles in Guyana that can transmit. Vincent's question on the swollen genitals would suggest ruling out Wucheria bancrofti, but in my literature search, it was not clear if Brugia malayi and Brugia timuri are prevalent in Guyana, so perhaps it could still be W. bancrofti. On to treatment. Diethylcarbamazine looks to be the drug of choice. An alternative could be ivermectin to kill the microfilaria, with doxycycline to take care of the adults. For the open wound, antibiotic to clear the infection. Bill is in Buffalo, where it's very cold and there is lots of snow, but no risk of mosquito bites.
Right.
And the last one is from Jesse. I really enjoyed TWiP, both the entertaining banter and the science. I particularly enjoyed Dick's expositions on the myriad parasitic ecologies. Daniel's clinical expertise is a welcome compliment to Vincent and Dick's tributes to Abbott and Costello. Thanks, Vincent, for supplying Dick with a new microphone and coaching his microphone technique. As a turnabout, a video of Dick teaching Vincent how to cast with a fly rod, while unrelated to parasitism, would make for bemused viewing.
Oh, great idea.
All right, we'll do that next summer. Not now. I'm not fishing now.
No, we're not going to do that.
Here's my take at 83. From perusing my wife's medical references, I'll venture that the Guianan gentleman has lymphatic filariasis. Probably been infected for many years. Lack of genital involvement, an ulcer, Brugia malayi. I don't recall Daniel's physical and exam mentioning enlarged popliteal lymph nodes, but I imagine they were present. Look forward to listening to your next episode. Thanks again for a great podcast.
How about them apples?
Yeah, that's great. Thank you, everyone, because this was a good response, right?
You bet.
I don't know why it wasn't last time, but
I think there was timing again, but this time it was also a very short.
It's been a few days.
Maybe this
Right.
case was just so great.
It's compelling.
It was compelling.
That's right. Or if you ask for it, they will give you what you ask for. So if we, at the end of every show, say, please send in your guesses.
we do, Dickson.
Yeah, no, I know we do, of course. But maybe
So,
this time
Daniel,
they were listening.
The call to action.
What is the diagnosis?
So
do you
do we
want?
get to brainstorm first?
Yes, let's. Why don't you guys brainstorm a little, and then I'll go.
Well, I heard a lot of correct answers according to what I would have said it is. I would have said Wucheria bancrofti.
Why?
Because it's endemic to that region of South America, first of all. Brugia timori is only found on the island of Timor, which is in Indonesia. And Brugia malayi is not distributed throughout South America. It's primarily an Asian and South Asian distribution. So as a result, I would say that if he has lymphatic filariasis, it's due to Wucheria bancrofti. That's what I would say. But, of course, I would delay that diagnosis in favor of doing a KNOTT test, which I presume we're going to hear about today because we get to ask for more information now, don't we?
You do get to ask for more information. So you're going with Wucheria bancrofti?
Yeah, I am. And if I were going to do a KNOTT test, a NOT test,
Okay.
I would certainly draw blood at night.
Okay.
Because there is a circadian rhythm to the...
And since you're a parasitologist,
I am a parasitologist.
I will force you into... Do you think this is an active infection, or do you think this
question.
is a chronic?
Well, a lot of the swelling that's due to lymphatic filariasis is due to dead worms that then calcify, that then block that lymphatic drainage. And if you have enough blockage, of course, then you start to accumulate lymph below the blockage. And so what we see here is a gentleman with a long-term chronic infection of what may prove to be Wucheria bancrofti. There are other ways of telling too, by the way, but the best way is to locate a diagnostic stage that you can rely on, like a microfilaria. It sounds like he's had this for a very long time, and it's possible that all the worms are dead. Mm-hmm. And if they're all dead, then the only way to really find out is to biopsy at the level of the block and see if you can locate calcified lymphatics with pieces of dead worm. And the cuticle of these worms tend to survive beyond the calcification stage, so We have a saying in parasitology that is this. I inherited this from Harold W. Brown in his day, and it's true then, and it is true today. When they die, they calcify.
When they die, they calcify.
Yeah, so that's one way to look at it. But another way to look at it is with regards to Trichinella, which lives in a nerve cell inside the muscle tissue. When they calcify, they die. Mm-hmm. So it's the opposite. So in some cases, when they die, they calcify. And in other cases, when they calcify, then it kills the worm inside of that calcification. So those are my, just random thoughts.
Okay,
based on what.
Well, I thought initially, when we recorded that it was guinea worm.
Okay.
Because
dracunculus rather than
dracunculus, because of the wound.
Yeah.
But when you mentioned the swelling of the extremities, then it was clear that it was lymphatic filariasis to me. But I didn't know which organism. Right. It sounds like Wucheria is the right thing, right?
Wucheria.
Wucheria?
Yeah, named after Wucher. That was the man's name who discovered this, and Bancroft was the other
Wucheria. I had a lot of trouble on our episode getting
you did.
that right.
I remember that. So Wucher was the guy who discovered it.
What's his name? Benjamin?
No, I'll get his picture for you. We can post it on the website if this turns out to be the case.
Wucheria.
Wucheria was one of them, and Bancroft was the other, and they combined together to describe this one.
I pronounced it incorrectly throughout all of these
letters.
Not a worry. Wucheria Bancrofti.
Bingo.
Why didn't you correct me, dude?
W. Bancrofti. Because I enjoy watching you make mistakes. You watch me do that all the time, and you don't correct me, so why should I?
Okay, so you're going with
I corrected you about
Wucheria Bancrofti.
The asymptote. Yes, Wucheria Bancrofti.
There you go.
Okay. And Dickson, why would we not go with leishmania? Why are you going to knock that off your list?
Well, first of all, I think the elephantine-like skin apparition of a leg that's swollen up is not a fair sight for leishmania vectors. In this case, it'd be the sandflies. Other parts of the body might be a target for them, but I think this is not a well-vascularized tissue, and therefore it would not be a great place for a sandfly to light on and then try to suck out some blood from that very dense cornified tissue that you'd find at the surface area. So I think this probably turns out, because of the energy associated with these swellings and loss of sensation to some degree, patients exhibit the same kind of hazards as a patient suffering from lepromatous leprosy.
Okay.
And that is, you get these lack of sensations, and patients with leprosy sometimes put their hands in fire, for instance, and they suffer tremendous burn injuries as the result. And I think this man might have bumped into something unknowingly and caused a surface abrasion. I'm sorry, I meant abrasion, not an aberration.
That's the British pronunciation.
That
right?
was the British pronunciation. No, Vincent, you can correct me now if you'd like.
Okay, well, let me jump in.
So that's the we're randomizing here, so we're going to have some directional input. So I want some tests.
Okay.
What tests?
Well, remember, this man tells us I have limited resources, so whatever you're going to do next, keep it cheap. And so I'm going to respond to that. And what is the cheapest thing that we do? And the cheapest thing that we do is ask questions, get more history.
That's
right.
That's basically right.
And so I take that lead and I say, okay, well, the cheapest thing I could do is ask some more questions. And and then I remember when I was graduating from NYU School of Medicine. This was fortunately quite a while ago now.
Yeah.
And I had hair at that point, more than I do now.
Right.
And we had our graduation ceremony at Carnegie Hall.
Really?
Wow.
Yes.
That's impressive.
That's pretty upscale.
The author of the book Night came and spoke to us. Eli Wiesel, am I pronouncing that correctly?
Yes.
And I had just finished. I'm just getting my MD degree. I have that arrogance that is common in that crowd.
Prior to your first day as a resident,
when suddenly it all washes away, and
Yes.
you realize that arrogance was poorly founded. And this gentleman gets up there.
Yeah.
And he's giving us words of wisdom, which at the time I didn't appreciate, but I have greatly appreciated through the years.
Yeah.
And he says, when you are there with a patient and you're trying to figure out what's wrong with them, and you ask all these questions, don't forget to ask the patient, what do you think is going on? And I have been amazed through the years how often they know so much because they've been living with this.
Sure.
This man is from Guyana.
That's
He's
right.
seen other people with similar things. And so
We've been to a clinic for this.
yeah. So I ask
They
him.
do have them.
I say, so what have you got?
Right. Why keep us in the dark?
And he says, oh, I've got Bigfoot. Bigfoot. And I say, other people? Oh, yes, there's this woman. Oh, she has the worst case of Bigfoot. And he goes on to tell me all about this.
Nice.
And I say, well, why do you, and you can Google Bigfoot Guyana. And what is going to come up, but...
Make sure you don't type out Bigfoot, Oregon. You'll get something completely different.
Yes, but Wucheria bancroftis. And then he starts telling me why does he have it? Well, he doesn't have it because he was been bit by a mosquito.
Right.
He has it because the government officials in Guyana are corrupt, and there's no longer this universal treatment program that they had when he was younger. When he was younger,
got it.
people didn't get this, and then they stopped giving out the treatment, and they used to come around once or twice a year, depending, and everyone would get treated. Nobody would get this.
Right.
And so this man, 20 plus years ago, because according to him, the corrupt officials,
Right.
you start seeing he's not the only one in his community who gets Bigfoot.
Of course not.
And he gets it. This woman gets it so horribly that she's got to wear dresses that come down so far that
Right.
nobody can see the Bigfoot.
Right.
And
Which might protect her, by the way, from leishmaniasis.
it may
We don't
actually.
know.
Yeah, it may actually. And then I get the whole story. Have you gone? Have you seen somebody?
Right.
Have they diagnosed you?
Right.
And yes, previously he was diagnosed, and we're going to let Dickson jump in and talk a little bit about. But as our listeners wrote in, this is a disease that if you want to diagnose the active form, when there actually are adults in these, I mean, it's kind of horrible, but these nests, there's actually nests that you can see under ultrasound of these squirming. Now, we tend to talk about the scrotal ultrasound if they get into the scrotum.
That's true.
And Vincent asked a bit
Or the
about that.
lymphatics of the scrotum.
Or, and then they'll be sitting and you can actually see them.
Yeah, inguinal.
And they're producing these microfilaria. And what are the microfilaria in the blood? I think one of our listeners put in at night.
Right.
During medical school, I was actually looking at a number of different research projects, and one of them was to study this in India. The only problem was, all the blood draws had to be done at 2 a.m. Now, that probably would have been fine, right? Because I could have stayed on, on our time zone and gone to India and just never seen daylight, and it would have been perfect. But that actually would make making a diagnosis of an active case here in the U.S. pretty hard. Where are you going to find a parasitologist who's going to be willing to look at stuff in the middle of the night? Well, you just need to collect the blood.
That's
in
all
the middle
you
of the
need
night.
to do.
You don't need the parasitologist.
You need a couple
to
of
be
mils
awake.
of blood. That's correct.
But that is important. And the people, if they come from an area with the nocturnal periodicity, remember, if they hop on a plane and they fly here straight from India, find out when 2 a.m. in India is in our time, because it's going to actually take a while before the periodicity of the microfilaria in the blood changes to local time. It eventually will.
Right. So, interesting. Go ahead.
So in
I have
this
so
case,
much to talk about here. I'm bursting with information.
So in this case, many years before the person was diagnosed, he was treated, and we'll talk about different treatment options. Unfortunately, you get scarring of the lymphatics. And what this man, when we look, we can see all the dark coloration. We can see all the elephantiasis changes, which hopefully we described reasonably well. Maybe that's why everyone got this right, or so many people got this right. But this man probably has chronic changes. And when you look at the wound, now I ask a few more questions about this wound, because, I want to know, was this suddenly you saw a blister and it was burning and you needed to put it in the water? I mean, is that what happened? And this is just, it never healed? Or did this... And so that would be our dracunculus story, right? And usually that's going to be, the skin around it is fine. I mean, the skin doesn't... You're not going to have chronic elephantiasis changes from dracunculus. No. That's going to... And so now the other, what about the leishmaniasis, right? That's going to start off as like a papule. Oh, I got a bug bite. I scratched it. It enlarges. And that would be your classical there. You're not going to have my whole leg all the way up with all these changes. So that story wouldn't be as consistent. then I get more of the history. So you've got this wound. What's going on there? And he periodically gets wounds. He periodically gets breaks in the skin. And this is just one area where he got a break in the skin.
Sure.
At one point, it was purulent, nasty, draining. And now, as far as he's concerned, it's hasn't completely healed and doesn't look so great to the sun, but in his context is not.
Yeah.
So we don't necessarily need to treat him for active parasites,
right.
But there is going to be a lot, as some of our listeners are going to try to focus on how to treat the lymphatic obstruction. A lot of good wound and hygienic care to this area, which really is at risk for trauma, skin breaks, bacterial superinfections.
Sure.
This is Wucheria bancrofti.
Cool.
And the expensive tests, which probably weren't that expensive in Guyana, had been done there.
Right.
And let's talk about the NOT test, the KNOTT.
I would love to do that
test. So we'll give Dickson
even
the stage.
Before that, though. No. This is a conversation.
Okay.
Come on. I will do this Socratically. I will ask questions, and then you give me the.
And you sort of look like Socrates, doesn't he? Actually,
would you please pass that cup of hemlock over here? I'm thirsty. So. And I always accused you of beginning sentences with the word "so," and I've picked up that habit, and it's not such a bad habit, actually. So...
I've got how to do it right here.
Exactly right.
The
NOT test is there. The modified NOT test.
Show you how to do it.
It's a way of concentrating microfilaria out of blood and then finding the one or two microfilaria that you might use as your identifying feature for this particular infection. And as I recall, the microfilaria of this particular species has an extended sheath around the second stage larva from the first stage molt, which didn't actually complete itself. So this worm is sheathed in a sheath that you can actually see if you stain the pellet that you get from the NOT test technology with Giemsa. You make a thick smear of this, right, on a slide, and all the blood cells are laked open so that you don't get to see those. The white cells are still hanging around in terms of their nuclei. And then you see these sinuous little forms that you can identify using a high-power microscope. And then when you switch to oil, you can look at the tail of the worm, and you can see whether there's a space between the worm and what looks like another transparent sheet of something over the worm, and that's the cuticle that hasn't been shed. That's pathognomonic for that particular infection. And there are other characteristics too. But so what I really wanted to talk about was, why do you think this parasite doesn't appear in the blood during the daytime?
How is it? Should we? Because remember last time you asked a why question, Vincent, and he jumped on you. So I'm gonna. How is it?
Where do they go during the daytime then? If they're not in the peripheral blood, where the heck are they?
Yeah, and I think the other is the issue. This is a vector-borne disease, as one of our listeners pointed out.
It is a big vector-borne disease.
And
big deal.
And this is actually going to be a great reason why people in Africa are interested in bed nets. Now, if you tell people, we talked about last time, we tell people, hey, you're going to get malaria if you don't use bed nets. So we talked about, most adults are like, yeah, it's a common cold. It's malaria. Right. Who dies of malaria? I get malaria every year, and it doesn't bother me. But if you get Wucheria bancrofti, it's going to make your genitalia swell, that is a problem. So men are very upset about that. And this would be the Vincent thing that he asked about. They used to make a big deal about this, and the most recent stuff I've read has said maybe you shouldn't make a big deal. But it used to be that different forms of lymphatic filariasis, as far as the geography, would be more or less likely to affect the genitals. And in sub-Saharan Africa, it is very common to affect the genitals. The idea was, and this has been called into question, but I'm not sure, is that in South American lymphatic filariasis, you tend to get less genital involvement. I'm not sure how true that is, but that was the classic teaching. And so if you're in Africa and somebody has elephantiasis of the leg and they're a male, then you may see the scrotal enlargement, which is very upsetting to people. Some things in some people's mind are worse than death. Dying, we're all going to die, but we don't necessarily need to have this scrotal enlargement. Right. So it's helped with the bednet campaigns, because one of the vectors can be the same vector as malaria. It can be Anopheles.
This is true. And
it can bite at night. And so the bed nets, which they get the men to buy into because, hey, it's going to prevent this horrible thing from happening. By the way, you're also going to protect you from malaria, and it's like the flu vaccine. If we can get less people infected, we have less ability to spread this disease.
Yes. So
well, that's my guess. What's the purpose of a nighttime parasitemia? Can I call it that?
Sure.
It has evolved that way because mosquitoes,
it's parasites in the blood.
Mosquitoes bite at night, right?
Well, you're sort of anthropomorphizing mosquitoes now.
No, it's evolved that way.
Darwinian selection.
It's been selected.
You betcha. Yeah. So there's something called non-periodic.
But they also bite. Don't bite. Mosquitoes bite all day.
There
right?
are two. No, some of them do, but many of them bite just at dusk. Or they bite late at night.
What's the mosquito here? Culex, right?
Culex species, but you can have many, many different species of Culex. You know how many species of mosquitoes? There are like 1,600 different mosquito species, I think. And many of them are Culecine. And of the Culecine mosquitoes, many are Culex. So Culex...
Have you seen a Culecine?
Quinquefasciatus is the one that I remember the best as the archetypal Culex mosquito species in India, which is primarily the vector for periodic filariasis. So I'm calling it periodic because there is another form of this, which is also Wuchereria bancrofti, which is not periodic. And Brugia malayi is certainly not periodic. Okay, so it has no periodicity whatsoever. But the reason why Wuchereria bancrofti has a periodicity is it's related to the physiology of the microfilariae. So you'll say, well, how can a worm disappear from the peripheral blood during the daytime and suddenly appear at night? What's going on here? So they found out. They have animal models for this. And they found out that during the daytime, the microfilaria hunker down in the blood that supplies the lungs with CO2 exchange and oxygen exchange. You can find all of the microfilaria in the pulmonary circulation at that point, in the capillaries. And the moment the O2-CO2 ratio changes when you go to sleep, and the O2 levels drop, and the CO2 levels go up as your metabolism slows down, as you start to...
What you're not explaining?
Then
you're not
the physiology of the
basis for that, though. You're not
do we know the genetics of the microfilarial responses to CO2 and O2? We don't, but we do know that they detect the difference, and they prefer a lower oxygen level in the blood to circulate. That's the way they're.
What happens if there's too much O2? They die.
Well, no, they don't have. When there's too much O2, they seek refuge around the lungs. Mm-hmm. Maybe that's where the CO2 concentration is the highest. Maybe they're attracted to CO2. Who knows?
Okay. Sounds good.
And one of the ways
I'm just guessing.
One of the ways that our listeners might remember this, right? People are, they hear all this stuff, and how do they put it together? So we know lymphatic filariasis, you can get the elephantiasis, right? You become the elephant man with the leg swelling. You also can get, in men, scrotal swelling. In women, it can affect the breast-dependent tissue.
That's right.
Now, there also is a syndrome called tropical pulmonary eosinophilia.
Bingo.
Where the microfilaria are going to the lungs, as we discussed, in this dormancy, this waiting phase. That's been selected for so that they'll be in the blood when the vector is biting. And people who have a robust immune system can actually develop this, we call it tropical pulmonary eosinophilia. Very high levels of eosinophils and a really robust immune reaction to the microfilaria in the pulmonary.
That's right. And it causes respiratory difficulties as well. So during the Second World War...
And that was what I was supposed to be studying, actually, if I was going to go to India.
Right. Oh, I see. If you were a soldier during the Second World War in the South Pacific theater of operation, you came home positive for Wuchereria bancrofti. Your blood had microfilaria in it, and yet we had not one single recorded case of lymphatic filariasis in the clinical form that you can see in this gentleman. Not one case.
Why is that?
That's exactly what I wanted you to ask.
Do you know the answer?
I do. Of course I do. So the clinical syndrome is not induced by a single pair of worms or 10 worms or even maybe 20 or 30 worms. It takes many more than that to block up a lymphatic drainage.
Yeah.
And so it's a long-term chronic exposure that gives you these syndromes. But that didn't stop the Japanese airplanes from distributing leaflets over occupied territory showing gigantic scrota of males saying, you are all being infected by this parasite, give up, there's no hope, you're all going to end up looking like this. That was one of their propaganda techniques to try to get the people to give up. And of course,
that's I remember this story. That's scary stuff.
That's very, very scary stuff. And you're tempted to, no, it must be like Tokyo Rose. She's, but maybe there's an element of truth to this. So every one of those troops got the knot test at the end of their tour of duty when they went home. And of course, now can you give blood? Can you get this infection by transfusion? And the answer, of course, is...
I'm gonna let you answer that.
No. I want the the answer is no, because you have to get bitten by the next stage of the infection that the mosquito acquires develops in the flight wing muscles of the mosquito. It crawls out as a third stage larva, and when the mosquito is biting, this worm crawls out onto the surface of your skin, and the mosquito doesn't even inject it into the bite wound. The larva has to crawl into the bite wound after the mosquito withdraws its proboscis. So people have thought about, well, if you're in the military and you're exposed to this, maybe we can come up with a cream or something which you can rub on the exposed parts of your body, so that when you're bitten by a mosquito that carries lymphatic filariasis and the larva crawls out onto the surface of the skin before the mosquito withdraws its mouth parts, it dies because it comes in contact with an anti-nematode kind of thing, like topical ivermectin or something of that sort. I'm not sure how far that idea has gotten. But
yeah, I mean, the thing you bring up, which is interesting, is that this also affects how efficient the vector is, because the humidity. There's actually going to be a certain amount of time. So if it's really, really dry, those larvae are not going to be able to penetrate before basically they desiccate and die. Yeah. So you need not only the vector and the microfilariae and the proper stage
everything
larvae,
has to come
but
together.
You actually need enough moisture to keep these
we
things alive.
We have a question in the back.
Victor.
Victor.
Yes.
Why didn't any of the returning servicemen develop symptomatic disease?
Because they weren't exposed enough.
What do you mean
They
enough?
didn't spend enough time.
Over and over?
Yeah.
You mean you get multiple
years.
infections?
You need to be there for years. So here's...
multiple bites and delivery of these forms.
That's correct.
Yeah, you need a lot of worms to die.
Well, the next thing you need is these worms live a long time. How long do Wuchereria bancrofti adults live? And so the estimated lifespan of an adult is about 10 to 15 years, during which time it actually inhibits the division of endothelial cells in the lymphatics. It inhibits the division of cells. So there's an antimitotic effect that this worm secretes into its environment to allow the vessels to remain dilated, because that's where their food is coming from.
You can't secrete an effect.
No, they secrete something that affects the division
of
the... That's what I'm
just making sure you're clear.
Thank you.
Hey, Dickson.
Yeah.
So if you get bitten once in this, you get infected, those worms do not multiply.
Wait, you get infected. You get bitten once, you might only get a single worm. It's either a male or a female. So at that point,
let's say you get bitten
worries.
twice and you have a male and a female.
But remember, the microfilariae are not causing the disease. The adults are. So you're right. So you get bitten, you have one adult.
That's it.
Even if it dies and calcifies, you're not going to have anything.
If it dies and calcifies. But let's say you get bitten twice, male and female, and they reproduce. Could they eventually make enough worms to cause disease? No.
No, because the microfilariae are not causing the disease. The adults are, you see. So it's the adult worms that are causing the disease.
Why can't they become adults?
I just told you, because they have to go through an insect vector in order to mature to the next stage. So,
all right.
So,
and that's the thing with a lot of parasites. There's very few parasites that in a human can complete their whole cycle and create more. Most of these require, it's got to go out, it's got to sit in the soil, it's got to be in a vector, then it comes back.
But I'm sure that you will come up with some cases of the exceptions to that rule.
There definitely are some exceptions,
and we would love to talk about them because they're exciting to talk about in terms of clinical.
So did you treat this gentleman?
So
yeah, what happened?
So this gentleman had been treated, and let's talk about treatment, because the treatment varies.
He doesn't look like he's been treated to me.
Whether you're in South America or whether you're in Africa
or India,
and part of it depends what other parasites might be around. Now, there's a drug called DEC.
Yes.
Diethylcarbamazine.
Correct.
Pronounce that correctly?
One of our listeners mentioned that.
And that was mentioned by one of our listeners.
That's right.
And that's a treatment often used in South America. And actually, there are studies looking at, we add iodine to the salt. They want to add DEC to the salt in South America just to get it out there.
Please don't do that.
Now, that would be a problem
Yeah.
in other parts of the world.
Right.
And because TEC works on other organisms. And so in Africa, Dickson, what would happen if somebody had a second organism?
Well, I'm not going to speculate on that level because I don't know the answer, although...
So Onchocerca is where I'm going with this.
I see your point. Okay,
so areas
other filarial...
areas where they have river blindness, right? Onchocerca.
Yeah, West Africa primarily.
If the microfilaria level from that infection is above a certain level and you give them DEC,
Right.
DEC kills everything.
Yes.
And is very effective. And it will lead to...
Except the adults.
Such an immune response
Right.
that it could actually kill people who were co-infected.
You know what? Giving small amounts is called... There's a specific test with the name of a
there
gentleman.
There's
He's
a
Italian.
reaction.
He's an Italian. His name is a Mazzatti.
Yes.
The Mazzatti test.
And
you give a little DEC to see if they've got it, and you get this huge skin rash. Up! You've got onchocerciasis.
So we don't do that anymore because some people actually more than a skin rash, they would actually die, and we frown on that. In the modern world, we frown on that. But that used to be okay, I guess. So the Mazzatti test is now relegated to the history books.
Exactly.
But that reaction is why you don't have to worry in South America where they don't have, except for I think there's like 3 isolated pockets in Venezuela. So maybe you'd
through
work.
Central America, they've got it.
But
there it is, the Mazzatti reaction.
Mazzatti.
Yeah.
They made a... Why couldn't they have named a car after him?
So this man had previously been treated with that. But the other option would be ivermectin. So when there are mass treatment campaigns, you might see ivermectin being used.
And what is that treating?
I have that. I
you can't look.
I made a little note.
No fair looking.
Well, I think the thing is, in my little note, I make a comment. The thing, ivermectin only kills the microfilaria, not the adult worm. But I think you are making the comment that the adult worm is also affected by ivermectin. Are you?
No. Ivermectin limits the reproductive capacity of the adults.
And that is true, but doesn't actually kill them.
No.
But it does limit
not
their
kill them.
reproductive
That's right.
capacity.
So, but the problem is that these are all long-lived adults, either the Onchocerca volvulus in West Africa and in Central America or Wuchereria. You have to give this drug for a long time if you're trying to stop transmission.
Yeah, and we're talking 10, 15, even up to maybe 18 years.
Right.
I mean, so this is a long commitment.
So in West Africa, and I guess we'll get to that one later, with river blindness, you have to treat once a year for, let's say, 20 years.
Yes.
But they did. They persevered, and they eliminated it from many, many, many places in West Africa as the result. So it's quite a remarkable...
So searching for Bigfoot Guyana reveals that this is a big deal in Guyana.
A very big deal.
There's a website by the Inter-American Development Bank where they talk about attempts to eradicate, to control mosquitoes.
Yeah.
There's 68,000 people, 9% of the population are at risk for infection.
Here's my next question then.
Yes.
Where do you think Wuchereria bancrofti came from in South America and in Central America and throughout the Caribbean and at one time in Charleston, South Carolina?
What do you mean, where did it come from?
Where did it come from?
I was gonna.
What country?
I was thinking of the slave trade.
Correct.
From where?
That's where we got.
Correct.
That's where we
Africa.
got everything.
It came from Africa.
That is our just punishment.
Is that how malaria? Is that how yellow fever came to the U.S. also?
I'm not sure, but it might have.
Yes.
I think you mentioned that on a previous TWiP.
I believe it did, and as a result, so there were a lot of slaves that didn't escape in the United States. Some did, but most didn't. But in South America, lots of them escaped, and they ran into the jungles, and they established populations quite apart from their captors in Africa. And they brought their parasites with them, of course. And so that's how schistosomiasis got to South America as well. I'm sure that's how yellow fever got there, and I'm sure that's how Aedes aegypti got there. And certainly the vector for malaria, the biggest one, is Anopheles gambii, and that got there at the same way. But they, you know what?
Well, why would slave trade bring a mosquito?
Well, the boats.
The boats are infected?
The boats had mosquitoes all the time.
Yeah, there were no
They had life cycles going on.
There were no
the boats.
mosquitoes.
So the
for
ballast.
instance,
Imagine how awful that
like
must
in the water
Yeah.
in the bottom of the boat,
Of
right?
course,
The
yeah.
ballast.
Captain Cook introduced mosquitoes to the Hawaiian Islands. The Hawaiian Islands had no mosquitoes. Can you
That's
imagine how
correct.
wonderful that was? And then Captain Cook shows up and in the bilge water.
Yeah,
I
bilge
know.
water. I said
So no
ballast,
wonder
but it's bilge
they
water.
killed him.
And five
They didn't know. They didn't even know then.
5 genera of birds in Hawaii were extincted by bird malaria.
Driven
as a
to
result.
extinction.
Is that
Driven
is
to
that a word,
extinction.
extincted?
No.
You made a verb. You verbed it. Dickson, Alan Dove doesn't like that.
They were extinctualized.
Extinctualized. Driven to extinction.
Driven.
The man has good grammar.
Well, not bad for an NYU graduate.
So he was treated. So you didn't treat him.
He
anymore.
was treated. So at this point, we didn't treat him for it, and he went back to Guyana.
What do you
they're
do
very
with him?
familiar.
Though? You tell his wife that he's okay, basically, because that's who wants to know, or his son.
His
whatever.
son. We tell his son that this is fine. You need to keep track of this, and the man knows better than most people here. He's had this for 20 years. He gets repeated infections, and he knows that when he gets an infection, he has to do dressing changes, and he needs to be careful not to strike this and break the skin and get injuries the way a Charcot foot in a diabetic, or the way the lepromatous lesions.
So there's nothing to be done about this lesion.
Yeah, at this
right?
point he's already gotten his medicine, and then down there in Guyana they've already killed the adult worms, and now it's a question of the treatment of the chronic.
If someone came to you with a lot of money and wanted to take care of this lesion, could there be some kind of plastic surgery done?
Now there's a surgeon here at Columbia, because I actually won't disclose who it is, but there's a researcher I know here at Columbia who has lymphatic problems from a prior infection.
Really?
And there's a surgeon here who actually does these lymphatic bypasses to try to restore the lymphatic supply.
Sure.
And it's tragic how many people suffer from this in the world.
No question. And in India, the microsurgery that resulted in the bypass operations was perfected there. Because they have clinics. If you go online, you can see scrotum clinics where men will be sitting at a bench, and it's just appalling to see the extent of their disease. It's quite an amazing thing.
And they can do bypass to reduce the swelling.
Yeah, they can not to reduce it. They can allow the lymph to go past the blockage that occurs after the adults calcify. So
a lot of these campaigns. When I was in Malawi last winter, there's this "Let's Make Malawi Filarial Free."
Right.
And part of the way they get buy-in is they, even though it's expensive, they get surgeons to go around to these villages, and the men who may have sort of leadership positions. You say, if you guys engage in our filariasis-free campaign with these treatments, we will have a surgeon come and fix your issue, and they'll get scrotal surgery.
Yep.
And it helps with getting buy-in to have that, to instead of just say, oh, it's too late for you, to be able to jump in and do something. And this man, fortunately, just the leg, he didn't have the scrotal involvement.
When we had a larger course here at Columbia's medical school that I used to teach, as part of the laboratory demonstrations, I had 1/4 of a scrotum removed from a gentleman from the Virgin Islands. That was the talk of the town, so to speak, for the medical students, because they just couldn't believe how much tissue there was there. I mean, the jar for this was clearly the size of a very large stew pot, with a lid this was a bell jar with a vacuum release on it, and inside of that was a preserved portion of this man's scrotum that had been excised after they did the bypass operation. And by the way, most of that tissue, you'd say, well, what is all that tissue? It's pretty dense. You can indent your finger into it, and it takes a long time to come back, but nonetheless, it's heavy and it's dense. So what do you think most of the tissue is inside? And it turns out to be smooth muscle. Mostly it's smooth muscle. And it's quite remarkable. I guess it derives from arteries or arterioles that are affected by the amount of lymphatic material that accumulates. It's food for these cells, and they just multiply like crazy. And so that's where this comes from. It's not cellulite per se, like accumulation of fluid. This is solid tissue that the lymph turns out to be a growth medium for the interstitial tissues that surround all these organs.
All right,
great case, by the way. Great case.
Now, do you think we have time to discuss our article?
We can do it. This is not a long article.
It's not a long article, and it's
but it illustrates some interesting principles.
I know we have time.
Okay, great.
This was actually suggested to us by Jesse, who wrote Doctor's TWiP. I liked the discussion of the interaction of bacteria and leishmania in sandfly guts. It was very interesting. So he had originally suggested that paper. Good. Here's another suggestion. So he suggested this paper. And Jesse, by the way, has a podcast of his own called Bacteriophiles.
Really? Which
is an ASM-sponsored podcast. You can find it at microbeworld.org just microbeworld.org, and you'll find Bacteriophiles on the right. The name of the paper is Delivery of a Functional Anti-Trypanosome nanobody in different tsetse fly tissues via a bacterial symbiont, Sodalus glassinidius. I don't know where you put the accent there. Right. What would you say? Glassinidius?
Glassinidius.
Are you familiar with Sodalus, Dickson?
Not even a little bit.
Are you, Daniel?
No, not really.
No?
I was familiar with Glossina, which is the species name for the tsetse flies.
This is from Belgium, a group there, and the others are Devoot.
I'm sorry, it was the genus name, not the species.
Devoot, Callion, de Ridder, den Abele. Yes, lots of mispronunciations. I'm sorry, but
et al. This is an et al. Paper.
It's only 1, 2, 3, 4 authors, but I mispronounced all of them. So Dickson, do we need a treatment for this?
Well, this is an unusual
trypanosomiasis
door to try to open with a control measure, because if we now backtrack a little bit to just remind everybody out there of the original episodes of TWiP, we covered the basics of African trypanosomiasis, and one of the main features is, of course, a crossword puzzle favorite word that always appears, and that's tsetse, T-S-E-T-S-E.
Great
right?
word.
And it's an easy one to remember.
Is it not tsetse?
It's not tsetse.
It's tsetse?
It's tsetse fly. So the tsetse
Do you agree
flies,
with that, Daniel?
Oh, I am not the pronunciation expert.
Well, I've heard it pronounced that way, so let's put it this way. I'm repeating a lot of other people that I've heard that were experts at this.
Okay, tsetse.
That told us how to say
the tsetse fly.
So the tsetse flies. So in Africa, you'd think, well, it's transmitted by the tsetse fly. There isn't one tsetse fly. There are whole bunches of them. And there are two kinds of trypanosomes. One is an East African trypanosomiasis. There's a West African trypanosomiasis. And when you look at the distribution maps, for instance, if you go to my website, Medical Ecology, and go to infectious diseases, and then go to vector-borne infectious diseases, you will get map distributions for the different species of tsetse flies. And so you have the Morsitan group and the Palpalis group and the Glossina groups. And you've got a tremendous number of varieties of tsetse flies. And they all have different biologies, all right? They all have different biologies because they locate in different places throughout Africa. So what if, I hope that all of those species has this same endosymbiont, because if they don't, then this will confound the control programs.
I already sensed a little bit of skepticism here.
Number two is the bigger problem of, even if it's successful, how would you get this into the tsetse fly populations? That's the biggest one.
Oh, you'd have to catch them all and inject them.
When I told my wife, she says, what are you going to be talking about today? And I told her, she says, and you get the little fly with little tweezers and you get a little syringe.
Intrathoracic.
It's not quite like that. It's not quite like that.
Dickson, we did a paper on TWiP where they injected microRNAs into ticks.
Oof.
They injected 10,000 ticks intrarectally.
Wow.
Oh, my gosh.
I don't even know how do you find the rectum on a tick? Oh, my gosh. Can you imagine how many? Whoops, sorry. We went right. Anyway, this is a very good point. There are lots of issues with this. However, it's an interesting approach.
It's fascinating. The fact that a nanobody.
What is a nanobody, Dickson?
Well, it's a single-chained.
It's not even. It's even more than that. It's even less than that.
Yes.
More is less.
So let's start with an antibody.
So an antibody has got 2 light chains and 2 heavy chains.
It looks like a Y, right?
It does.
And the tips of the Y.
That's right. I used to do this a lot with my hands.
That bind the antigen.
That's right. And the pocket. We knew a gentleman here by the name of Elvin Cabot, who was at the height of his career. He was describing the amino acid variations inside the binding site for antibody molecules, and there were 3 hypervariable regions, as I recall, for each of the chains, and 3 and the rest of it was non-variant.
Yeah.
And the antigen selection point was for the variable region. Okay, so that's where the J regions and all that stuff that comes together to produce the diversity index for all your antibody molecules comes from. And so what have they learned? And this is something that is semi-new to me. I mean, I know a little about this because I hear these things, but it only requires one of those chains in order to bind to the antigen.
Well, that's that's what an antibody is. So it turns out that camelids, right? Is that the word camelid?
That is it, camelids. So those are camels, alpacas,
llamas,
or llamas, right?
Guanacos.
The two L's.
Don't forget the
or
guanacos.
llamas. How do you pronounce that?
Depends on which country.
Yes. So the camelids.
It's llamas.
They make single chain antibodies.
Yes.
Get out of here.
Heavy chain only,
Stop
right?
it. Is this true?
Yeah.
So this is great evolutionarily, right? I mean, when did antibodies evolve? And you can go way back to...
This is a mammal. These are mammals
That's
you're talking about.
So you can go way back
Mammals?
to... Who has antibodies? So you don't have
Sharks,
antibodies.
teleosts.
Sharks. So we're going all the way back to sharks.
Correct.
And if you go back to the nurse shark, maybe this is kind of new stuff. The nurse shark, not only does the nurse shark have heavy and light chains, but the nurse shark also has these heavy chain antibodies, just a heavy chain, that function very similar to the camelid. So the camelid went for it, and actually you can trace it all the way back.
Camelids are sharks?
Well, they're
land
descended.
sharks?
They're just yes, exactly, and they're about as nasty, right? I mean,
We know land
they'll
sharks from
spit
Saturday
at
Night
you
Live.
yeah, so when these things evolved, in the shark, when these things were evolving, you had the ability to use heavy light chain complexes like we use, but they also had the ability to use heavy chain only binding.
Why didn't we also adopt that as another arm of our...
Can't ask a why question.
Yeah, look at it. He asked why again. He's terrible.
Did I?
You know,
He's-
you were
like
the
kick him
person.
out of
You were the person who introduced me,
I know, but
and you violated consistently.
Do as I do. Do as I say, not as I do.
So, do you think this is an early evolutionary stage of the antibody development, having a single chain?
I think that when they first evolved, there were the two options, and we still have the two options preserved to this day in the nurse shark. So there must be some advantage to keeping it, as the nurse shark did, or using it exclusively, as the camelids are.
So, having two binding sites on the Y's allows you to bind two sites, maybe cross-link. So maybe that is why we have them, because we
I
have to
always thought
cross-link.
of this as a pocket for the antigen to fit into.
You have two binding sites on the Y.
I know
right?
that. But
So
they're
you can-
complementary to each other.
They're the same binding site.
I know
Yeah, but
that. So you've
but
got
we
the
like-
hypervariable regions on each of the chains coming together, and the antigen is stuck tethered in the middle by
No,
these
not
three
necessarily.
binding sites.
You can imagine the surface of a bacterium or a virus. It's interacting with two of the same epitopes, and that's called cross-linking, and it may be needed.
Yeah, and this may be part of our
See,
attempts
I'm
to
looking
find...
at soluble antigen,
Yeah.
and you're looking at particulate antigen.
Yeah,
No, usually
we're-
our antibodies
Isn't
are
that
triggered by, as Vincent's saying, by having multiple of these called antigens or antibody targets. You want to have multiple ones because you're trying to always balance between recognition and immune response and not having autoimmunity.
But
not firing off when it's just one.
Yes, no, come on. I'm not that naive about this. I remember lecture after lecture after lecture using soluble antigens.
Yeah, but
of
that's
various
not all
sorts.
that there
No,
are.
I appreciate that. But
Yeah.
but every time they described it, they would
No, that's
show you
wrong.
the pocket.
That's wrong.
And they would put the antigen in the middle of the
That's
pocket.
only
and
one
say,
Y.
this is
There's one binding site on Y. You never put an antigen in between the Y. Never. If you look in the immunology books, the antigens are binding on the tips of the two Ys.
So what are the other two variable regions for then?
They make the combining site. They all comprise the combining site. Okay.
But in a single chain, you can bind all three then.
Single chain
On a
binds at the tip.
nanobody, you can bind all three. To
Anyway.
different
So
That's
anyway,
it. No, that's
the
fascinating,
great
fascinating.
biotechnology here
Yes.
is that when we try to screen antibodies that are going to neutralize HIV or whoever knows we want, it's really hard because you can screen in these phage display systems, allow you to screen like millions of things.
See your point.
You can screen just the heavy, you can screen just the light, but we really, for most human antibodies that we might use, we want to know the combination of heavy and light together. Now, if you have a binding here that is just a pure heavy
what you're
chain,
saying. I
you
hear what
can
you're saying.
screen the whole camelid binding repertoire and find what you want and engineer it, stick it, as they're doing here, into this gram-negative organism, have that gram-negative organism start producing this therapeutic antibody as they're using here in our
you used
flies.
an interesting word here, therapeutic, and I
Yes.
want to get to that
All
word
right,
too.
but before you
because-
do, the real key here, as Daniel has alluded to, if you want to make an antibody, a recombinant antibody, you have
Right.
to produce two heavy chains and two light chains. They have to assemble properly.
yeah, it's
it's
complicated.
challenging.
yeah, got it.
A nanobody or a heavy chain only, you just have to make one protein. It's very easy.
So those are trivalent.
So people got-
in your view, these are trivalent.
No, these are
Each
not.
one
These
is
are one
you
chain.
get it against a different antigenic determinant. Each of these binding sites on that nanobody is against one
No.
antigen, 2 antigen, 3 antigen?
No. It's the
Or is it
same.
all against the same antigen?
It's the same antigen. Each
So it's-
antibody is recognized. That's why we call them monoclonals. They recognize one
Yeah, but I'm
epitope.
not...
Yeah, no, I'll jump in on this. So we call them CDR 1, 2, and 3, complement determining region 1, 2, and 3.
Right.
And think of it as a pocket. Think of it as your hand, except we're going to take away the thumb and your pinky. And you want to hold something in your hand.
Yeah.
And each modification is going to improve that binding pocket.
Got
But
it.
that binding
That's
pocket
called
is going to be.
selection. Yeah.
But so each of those isn't that it's binding to a different place. It's that pocket is now
Getting
refined.
tighter and tighter.
It's all
Exactly.
part of one.
But it's one pocket.
Site.
One
It's one
pocket.
site,
You're just
not
So all
three.
3 of these comprise the pocket.
Yeah, you have to think in
Exactly.
3 dimensions,
So,
Dickson. I know
okay.
that's hard, but
Elvin, are you listening, Elvin?
we have structures now which show this. But people got very excited when they found these in camelids
They
because
should.
all of a sudden you could make reagents
Yeah.
very quickly, and people
Yeah.
use this for research purposes. You could
Okay.
fuse GFP to your antibody and have a
Okay.
reporter right away.
Bingo.
Now, what's cool here is that these are even shorter. These are just the combining site.
Mm-hmm.
It's not the whole heavy chain anymore.
So there's no FC fragment.
Nope, it's gone. Just the
Okay.
just the combining site is enough.
Okay.
That's why they call it a nanobody, which I don't think is a very good name. But
Well, then
someone
how does it work? I mean, how does it actually work?
it folds and makes the pocket.
No. But even if you combine with something, the fact that what
Well,
does it do then next?
Yeah,
What's the
that
next
is-
thing that it
yeah,
does?
that is a good
Well, that's
question.
a good question.
Since you take away this tail, so to speak,
Right.
we'll call it a tail. The tail has effector functions.
No kidding.
So in a lot of ways, you're focusing on something that is going to bind, and the actual act of binding is going to have a neutralization. And that's what we see in this paper is that they've created these nanobodies, and now we're going to have to think 3-dimensionally of our trypanosome. So
Right.
how do we describe a trypanosome? So think of it as long and flat. Can I describe it as long and flat?
You can just do anything you want.
Okay. Think of it as a narrow leaf, so to speak, and it has the ability to quiver. It has a tail, but the interesting part, and this could try
And an
to visualize.
undulating membrane, too. Don't forget that.
Well, that's the interesting thing. You take this tail,
Right.
and the tail comes into the body. So
Right.
think of a mouse that you stepped on. The tail, but then that tail actually continues all the way up the undulating to come up to the head and the mouth, and there's a pocket there, and that's where sort of the motor of this is. And the nanobodies,
Flagellar pocket.
the
It's called the flagellar
nanobodies
pocket.
go into that and plug up that hole and compromise the trypanosome.
So what
you
is
don't
their
need-
specificity?
So it's
VSG.
the specificity is for the VSVG, so this variable surface variant glycoprotein,
It's usually,
and it actually they're
VSG.
targeting
Remember
one that
VSG?
is sitting.
do you remember tsetse fly VSGs? No. This is a procyclic trypanosome. This is different.
This is
Yeah.
trypanosome VSG.
No, Vincent.
Yes, Dickson.
Vincent. Let me just tell you something
Bloodstream,
about the life cycle.
bloodstream
This is
trypanosomes.
not bloodstream.
That's what this antibody
It's
is
not bloodstream.
recognizing.
No, what are they? What
So
are
this
they
is
making
actually
this against?
in
They're
the
doing it
mid
in the tsetse
in the
fly.
midgut of our fly.
The tsetse fly doesn't have
Yeah.
any bloodstream trypanosomes. None. Zero
Just-
zip.
Fine, that's fine, but that's
No,
what
not
this
fine.
antibody
This
is
is like viral
directed.
replication. You have to pay attention.
This
now.
is what the antibody is directed
I
against.
know. So I'm asking another question.
What's your question?
It can't be against the VSG because VSGs are produced by the bloodstream form of trypanosome.
Can I read the sentence from the paper?
No, I know what this paper says, but I'm.
Nanobodies targeting epitopes of VSG abundantly present on the surface of bloodstreams have been identified, some of which exert activity, as Daniel said, by binding to the flagellar pocket. Okay? That's what these antibodies are directed against. Now, what's your problem?
My problem is that I spent about 4 years listening to Lex Vanderplug talk about this stuff.
Yeah.
And part of what he was talking about was that this is sort of an area of the trypanosome that doesn't have the VSG.
Well, it works. It kills them. It kills them. He was
Excuse me.
wrong. Look, I don't want to.
Stop.
This is not
Stop.
worth arguing
No,
about because
yes,
they
yes,
show
it is, because this pocket is where the organism gets its nutrition.
These are trypanolytic. Okay, they target VSG. I don't care what issues you have. It works.
Well, I'm just trying to envision this. That's all.
Well, you don't have to depend on what Lex told you. He may have lied to you.
Well, I'm stuck with this old knowledge that is now totally obsolete. Bring me up to speed.
Well, I don't know about speed, but all we know is that this antibody kills trypanosomes.
Because I know that somebody in your family also worked for a very famous trypanosomologist over at Rockefeller.
Yeah, my wife worked for George Cross.
George Cross, who made his living studying VSGs.
Yes, I know.
All right, he actually discovered them. But I'm skeptical of this pocket that you're talking about. I'm sure that's where the action is. That's where this antibody is active.
Well, it says some of these antibodies are active that way. There may be other mechanisms as well.
Because the transport mechanisms for feeding occur through this flagellar pocket. That's the only place that the organism can obtain nutrition. But this is a lytic antibody, you said?
Well, they call it a trypanolytic.
How does it lyse it without the FC fragment?
I don't know, because the FC would
Another
allow it
interesting
to
question.
would allow it to interact with complement, for example.
Yeah,
it could be the
so
basis.
How does this work?
I don't know. It says these antibodies interfere with the endocytic machinery in the pocket, so maybe they can't take up and they burst.
Okay, no, that's fair enough.
Which you're about to do now.
I'm just bursting with curiosity, basically. No, I really am, because I'd like to reformulate
Dickson,
my way of thinking about
may
this.
I be so bold as to suggest that you have read the paper and gone into the background?
I read this paper.
And this would answer the question.
No, well, I read this paper, but I didn't have time to look up all the background.
This is no excuse.
Of course it is
You're
an
serving
excuse.
our
I just
listeners.
got this.
I sent this to you 2 weeks ago.
No. I actually didn't have a record of this on my email, but that's okay. The point is that I need to learn more about the trypanosome biology now, because they're really attacking this organism in the vector.
Yeah, I agree with you, but that's not really the focus of this paper. The focus of this paper is putting the gene for this antibody in the symbiotic bacteria that live in the fly and seeing if that works.
Well, they did a proof of concept afterwards. They did that. That's
so
true.
these symbiotic bacteria, these sodalis, live within the fly.
They have another endosymbiont too, don't they? In the gut tract. It's not an endosymbiont. It's just a symbiont.
I'm sure they have many.
Do you know what it's called? It's called Wigglesworthia.
I was going to mention that. Did
you know who that man was?
There was a poem by, I have it on my wall there, V.B. Wigglesworth.
Wigglesworth was a very famous biologist that worked over in England. He was an entomologist.
Really?
Really. And he worked on tritomids, which is the South American form of the vector that transmits Chagas disease.
Yes. All right, so wait
a minute. He's going to read this poem about Wigglesworth now.
Okay.
I have to tell you this story. I'm sorry that...
No, this is a diversion that's worth listening to.
When I came to Columbia, my first office, this poem was stuck on a filing cabinet.
Really?
All right? And you can see it's all yellowed, and it's by John Updike. It's called V.B. Nimble, V.B. Quick. Oh. And it is so beautiful. I have to read it to you. V.B. Wigglesworth wakes at noon, washes, shaves, and very soon is at the lab. He reads his mail, swings a tadpole by the tail, undoes his coat, removes his hat, dips a spider in a vat of alkaline, phones the press, tells them he is FRS, subdivides 6 protocells, kills a rat by ringing bells, writes a treatise, edits 2 symposia on Will Man Do, gives a lecture, audits 3, has the sperm club in for tea, pensions off an aging spore, cracks a test tube, takes some pure science and applies it, finds his hat, adjusts it, pulls the blinds, instructs the jellyfish to spawn, and by 1 o'clock is gone.
That's a great poem.
Isn't it wonderful?
It's wonderful.
And I saw this, and I said, "Oh my God, this is just perfect." I wonder who was in my office who stuck. And
Did you know who Wigglesworth was?
So I didn't know, and then I read this paper, and they say Wigglesworthia.
Right.
It's an endosymbiont, or it's a.
It's named after him.
Bacteria in the gut of tsetse flies.
Yes.
How about that?
How about that?
I can stop right now.
He was as generic in his interest of biology as E.O. Wilson is. He was a world-famous biologist that lived in England.
So he worked on trypanosomes.
I'm not sure. No, I think he worked on the flies. He worked on the vector, and he worked on tritomid bugs.
This is just great. Yeah,
among many other things, as John Updike obviously has shown us.
The obligatory Wigglesworthia symbiont population that resides in the tsetse fly abdomen.
Right. So you see, I did read the paper. I just didn't understand it.
These are great flies.
They're wonderful.
I mean, I was actually,
they're fabulous.
I was actually saying to Dickson, I call it off mic. So apparently now my life is either on mic or off mic. And I was talking about one of the great things about infectious disease is you get to be interested in just almost about anything because it all relates in.
Right.
And this, you think about flies, the nasty things, you swat them, they're just a pest. But now this is a wonderful fly. This fly has eggs. It takes those eggs, and the eggs actually hatch in the uterus. This is a fly that's going to give live birth. You ready?
That's right.
It's a fly that live birth from a fly.
Right.
And then while this when the egg hatches in the uterus, then the mother fly is going to feed it with milk.
This is true.
There actually are almost like mammary glands. They're milk producing glands in the uterus, and then the little baby comes out.
Aww.
How many?
One at a time.
One at a time. And then it gets better. Now, normally you think of flies, they're buzzing around here and there. Now, these flies, they're like hawks. They actually go up in the top of a tree. They get really high. They have great vision, and now they're watching for prey.
They're fabulous.
How big are these flies?
The size of a housefly.
Yeah, I
They're
would
not
say
so
if
big.
you looked at the nail on
picture
your
them in
the
the
nail on your
parasitic
the nail on your pinky,
textbook.
they're about 20% or so sort of off from the size of a housefly.
Yeah.
And so they get up there, and what are they? They're hunting. They like big animals, right?
Very big.
And they like big animals that move. So if you're sitting there in your Land Cruiser, let's say you're visiting the game park, you're all sitting there in the Land Cruiser, windows are down, everything's fine. Now the Land Cruiser starts to move. The tsetse fly catches it and comes zooming in. Now the worst thing is you have a blue Land Rover, right? Because they love blue or black or dark. If you're naked in Africa, they probably won't bite any of us because we're pasty white guys. But if you put on a blue shirt, they'll bite right through that blue shirt. I mean, these are amazing.
Painful bite, too,
Yes.
by the way.
These are amazing animals.
There's a horsefly mouth part. So it's like scissors that it scissors its way into the blood.
And they even have a proboscis. They look like they're like a combo mosquito, hawk,
That's right.
loving mother fly.
Did you also learn that once they're near that large moving object, there's another set of environmental cues to let them know where they are?
Well, they also like smell, so don't cover yourself
Very
in
much
urine.
so. No,
They love the
Cow
smell
breath. They love cow breath.
They love cow breath and cow urine.
Correct.
Yes.
So they can actually sweep these flies. Now, the density of these flies in East and West Africa varies greatly. So in West Africa, they're called riverine species, and they all live along the rivers, and there are thousands per mile, maybe even more than that, per mile of riverbank. But they don't live far enough away from the river to prevent people from living close enough to the river, but not right on the river itself, and still avoid trypanosomiasis. In East Africa,
then there's 1 per square mile. I think is what they say.
One fly per square mile. 1 fly per square mile. So if you're out in a Land Rover watching zebras and gnus cavort among the elephants,
how do you know they're not horses?
All you have to do is kill that. All you have to do is kill that one tsetse fly, and then you've cleared your area.
In Africa, I always think of zebras. And yeah, if you kill that one fly, you've cleared a mile's worth of African veldt. So in East Africa, they have this fantastic way of controlling this. They have a truck, that's painted like a zebra.
Not a horse.
Behind it, they're carrying what I would call the Trojan horse for tsetse flies. It's a artificial cow on wheels that secretes cow breath like molecules, like carbon dioxide. No, really, it's carbon dioxide and methane, because cows are anaerobic, as you know, in their digestion. So their breath contains lots of methane. Out comes the CO2 and methane. It leaves a trail behind this, and then the source of the methane and carbon dioxide is a funnel, which leads into a small container, and then the small container leads into a very large container. So this is essentially it's a CO2 trap, but they're using cow breath to trap it.
And the flies go in and get trapped.
And then they get in and they can't come back out again because they can't find that little hole. And why would they want to come out? Because it's loaded with the pheromone to them that tells them who to bite, and yet they can't find their victims once they get inside. You can clear out 100 miles of veldt like this.
We have.
I think that's incredible.
I think you mentioned that on a previous episode, but it doesn't hurt to mention it again.
I love to retell these.
Dixon, do you know why they want to make antibodies in this Sodalis?
They want to clear the infection out of the tsetse flies.
But why is Sodalis a good choice?
Because it's an obligatory endosymbiont.
It's got to be there. You can culture it. That's important, right? If you couldn't culture it,
Dixon, is to others.
If you couldn't culture it, that would be an issue, right?
That would be horrible.
It's in the gut where the trypanosomes are, they say. So it's close. Right? And you can genetically modify it.
Bingo.
Right? So it's also it's restricted to the tsetse niche, so it's safe.
Right.
So they want to put the antibody gene for the this trypanolytic antibody, which you despise. Okay, it's fine.
No. I just need to be brought up to speed. That's all. Please.
And the idea would then to recolonize the tsetse flies with this
that's
antibody-producing
the toughest part.
bacterium.
There's the part
and then at some point you're going to release them, right?
Yes.
And you say, then a miracle happens.
And then a miracle happens. So they can make these bacteria produce the antibodies. That's not a problem.
Right.
And it doesn't seem to interfere with their replication, their division. And it's stable and so forth. That's important.
Relatively stable.
Not 100%.
The stability was a bit of a challenge.
It's a plasmid, right? It's not integrated into the bacterial
It's a plasmid.
genome,
And it has antibiotic resistance marker, which you use to select. So you have to grow the bacteria in the presence of the antibiotic that's on the plasmid. But even then, not all of them maintain it forever. They pass it a certain number of times.
Plasmids are acquired and lost, right? Through your neck of the woods, the virome of the world.
Yeah. They call this recombinant sodalis.
Can you acquire a plasmid through conjugation of bacteria?
Who?
I asked first.
Who can acquire it? Me?
No, can bacteria transfer plasmids
Of course.
via conjugation?
Yes. They do it all the time.
Okay.
As
They
well
don't
as
need a viral vector.
they start doing, as soon as you put bacteria together, they start swapping DNA.
That's ridiculous.
It's like sex.
Is it Joshua Letterberg, I guess?
Yeah,
Joshua
Joshua Letterberg.
Who described that?
Let's see this stability here. Yes, 94% of the population remained antibiotic resistant after 27 generations, which is 2 months. So it's losing the plasmid, which is not good to begin with.
No.
Apparently,
what if it was perfect? Okay, let's talk about what if this was a perfect experiment and they never lost their plasmids and
no, that's only one issue. That's only one issue.
No kidding.
Then they have to put these in the flies. Okay, they do intrathoracic inoculation.
That's not.
I suppose they put a needle in the chest.
Yeah,
right?
isn't that amazing? Put the needle, a tiny needle in the chest
Come on,
of
we
a
used to
fly.
do this all the time with the mosquitoes. It's very easy to do that.
You used to inject
It's a no-brainer.
mosquitoes?
It's a no-brainer. Yeah, we used to inject mosquitoes. Yes.
Wow.
Yes.
Well, how about a tick?
You have to go
Intrarectally.
That's tougher.
So here's where the problem rears its head.
Right.
Unless you clear out the sodalis in the fly, these guys don't take.
Right.
And they have to use an antibiotic, right?
Because they outcompete the ones
Probably.
that
So putting this antibody probably interferes in some way that we don't understand. And so they can colonize and they can show that the flies produce antibodies, but it's going to be a problem for release because this is not going to spread to other flies.
There are many problems with release. Okay. So I used to know someone up at the University of Massachusetts who had a tsetse fly colony. A tsetse fly colony. Imagine that. I mean, that's rare. And they maintained it because they were looking at the development of the trypanosome in the tsetse fly, not in the bloodstream. They were much more interested in what happens once it gets into the fly and where does it go and how does it reproduce and all this other stuff. So I learned through secondhand information, of course, how difficult it is to manipulate this particular organism. And even if everything went perfectly well with this article, with their research, by the way, if the plasmid sticks, if the bacteria infects, if the trypanosomes are killed.
Hey, they didn't do that.
I said if.
That would be a good experiment. Why do you think they didn't do that, Daniel?
Well, that's the next step, isn't it?
Yeah, they didn't do that experiment, which is they showed they make antibodies, and the antibodies working in an ELISA. Yeah. But, Dickson, what would you do to show that these antibodies would keep the flies trypanosome free? What is the experiment?
Well, you need to complete the life cycle.
How?
It's pretty easy, actually.
You would inject
Trypanosoma brucei infects a lot of mammals.
So you could do a mouse?
You can't do the West African form, but you can do the East African form.
Yeah, I would think to use the East African.
You can't use the West African form because they don't infect a wide range of animals.
You need volunteers, right, who are willing to...
No,
can you
be IRB
you could
challenges.
do it in pigs.
Can you infect
that's
mice?
actually interesting. There are pigs. You can do West Africa. You just restrict your...
Can you do mice?
Mice, you can do the mice in
So
East
you
Africa.
would have an infected mouse. You let the flies feed on them.
Keep going.
And then you ask if there's a difference in the trypanosome load in the fly with and without the antibody.
Or even then let the flies then go feed on a non-infected
transmission.
mouse.
That's correct.
Because that would be the ultimate. You're trying to block transmission.
But wait a minute. I've just told you something here. There's a tsetse fly colony possibility.
Where's that?
Anywhere, it doesn't matter. You can raise them in vitro. You can raise them out of the African veldt.
That's fine.
So Why are we bothering putting plasmid inside of a bacteria, an endosymbiont? Why don't we just take the trypanosome
genetically
itself?
engineer the tsetse fly to produce this nanobody?
It's another approach.
That was
There's another
my...
I don't know how easy it is to get genes into trypanosomes.
Well, we got the entire genome. If you go online, you can actually look at the entire genome of tsetse flies.
I guess the idea is you're going to put the gene into the tsetse fly germline so that all the tsetse flies.
Yeah, how would you do that?
So wait a minute. We have a retroviral person sitting right over here. You tell me.
Oh, you're pointing to me.
I am pointing
Yes.
to you. Come on, you're an expert at this stuff.
Yeah, actually, I mean, that was my thought, of course, right? Because that's like, well, why don't I just use my tools? Why are they using these other tools?
So what are your tools?
Well, using retroviruses to insert new genes. Actually, I'm working on humans, try to put them into the stem cells more efficiently. So can you put these into the stem cells of tsetse flies? Now, there is some work that started mainly in the 90s in mosquitoes. We're genetically engineered mosquitoes. And so what they're doing, really, I was sort of surprised how similar it is.
We had him on TWiP. That was Tony James, by the way. He was the very first person to ever manipulate a mosquito.
Yeah. And so...
And what did he end up doing? He used promoter sequences derived from Drosophila in order to get it to work, because that's how you had to do this.
Yeah, and if you look at the work where it's evolved, they're using... I mean, now maybe we're getting into TWiP and we talk about promoter sequencing, but they're actually putting in genes into mosquitoes, and then they're putting in the sequences that basically turn on production of that protein. But interesting enough, the retroviruses have these long terminal repeats.
They do.
The long terminal repeats may be enough to get some level of production of this gene. I have not seen as much work done with retroviral manipulation of tsetse flies, but why not? Why not?
Well,
we could probably get some tsetse fly.
You could probably find it, but the key would be to get it in the germline. That's not
Okay, so
that
what
easy.
are tsetse flies?
Tsetse.
Stop it. No, what's the other part of their word?
Fly.
Yes, thank you. Stop
They
right
fly.
there. Think of another fly.
Housefly.
Think of another fly.
Another fly? The reduvid bug.
That's not a fly, it's a bug.
Okay.
You just said bug, didn't you?
Are you thinking of Drosophila?
Vincent.
Drosophila?
Are there transposable elements in Drosophila?
Oh, yeah, sure.
Definitely.
Oh, really? Oh, is that right?
Yeah, one of them's
you
called
mean I knew
Gypsy.
something? Oh, yeah. Yeah.
It's
So what
called
if we
Gypsy.
what if we took the transposable elements from Drosophila? Oh, wait a minute. Is that a fly?
Drosophila?
What is the name of that?
Yeah, it's a fly.
What
It's
is
a
it
fruit
called?
fly.
Thank you.
Why are you
So
getting
now
so
we're getting
bitter,
somewhere.
Dixon? You're so bitter because
no, I'm
you...
being emphatic. Because the tsetse fly, the mosquito, the Drosophila, those are all dipterans.
Yeah.
So dipterans have a lot in common.
The key to getting genes into Drosophila was to use these transposons,
So
P or Gypsy. So you could do that,
manipulate
but
a transposon, put it into tsetses, and see what happens.
But here's the thing: you're right. You're absolutely right.
Thank
It's
you.
a lot easier to do this in bacteria that are endosymbionts. That's why they took this route.
And this is easy. I mean, to put a plasmid in a bacteria, you got your little cup of ice, you got your heat block.
I know.
You mix
I
it in
know.
there, you
I
bounce
know.
it between
I
the
know.
ice and the warm
I
block,
know.
and now it's in. It's quite something to engineer a targeted retroviral vector to put this into the germline.
The point is,
I'm
though,
glad you
once you
brought this up.
succeed, you don't have to worry about that
Dickson.
anymore.
This is because these have great names. So the Drosophila has an endogenous retrovirus called Gypsy.
There you go. Why is it called Gypsy, do you think?
Why do you think?
Because it keeps wandering.
Actually, it
It's a
does not
wandering-
it doesn't transpose.
It's a wandering gypsy.
It does not transpose.
Well, then why is it called gypsy?
That's a good question. Ask Steve Goff.
No, it's called gypsy because it jumps from chromosome to chromosome.
Gypsy is an endogenous retrovirus of Drosophila. It is stable and does not transpose in
Well, then
most
why is it
strains.
called-
I
in
don't
most strains-
know.
In most strains.
Maybe in one strain
The
it
one it was
transposed.
isolated did. Was the
All right,
exception.
that's enough. This is not going anywhere. What do you think?
I agree.
But it's an
I
interesting.
like the idea, but
I love the idea.
I love the idea. It's very
Thank you
creative.
for raising this because
Well, thank Jesse. He suggested.
no,
it.
Jesse is. This is number 2 for Jesse now. Maybe we should put him as an adjunct.
Yeah, tell them to keep sending us articles.
As it keeps
shall
sending
we?
us?
Shall we? Before we lose all our listeners,
have
before
to.
we max out,
No, we
like
need
the
another case.
megabyte-
We
I
need
have
a-
another case.
We need another case.
And
Yeah,
this
we need
is,
a case.
I'm moving up to a case which is going to be a bit more challenging.
And I don't think we should guess because the guy doesn't like that.
We're going to remain silent.
Well, you can ask lots
The
of questions,
wheels are
but
turning
mention
the whole
no
time.
mention no organisms, mention no parasites.
Nothing.
You could just ask as many questions as you want.
Like what new tests?
And then I'll even throw you a little bit at the end.
I don't know.
I love this case.
Come on.
All right, go
So,
for it.
It's November. A 32-year-old man reports that he has been having recurrent fevers up to 103 degrees for 3 months now, right? So it's November. He's been having them since early September. So September, October, now it's November. He's still having them. He reports he's got muscle and joint aches. Energy's not good. He's got it. Describes as nodule on the back of his left hand, his ring finger. He doesn't know if that's related, but by the way, I need to tell you about that. He has a cough productive of minimal brownish sputum. He gets headaches and blurred vision with occasional, he calls them blind spots and floaters. And I'm going to give you a little more history just because you might not ask this. Where's he from? He's from Missouri. And he lives in a house in Missouri with a bunch of his buddies. And you ask him a little bit more. He's a normal guy, likes to drink a whole bunch. Likes to do fun.
What does he do for a living?
Fun Missouri things. He works in an office. Does
Office
paperwork.
Worker. Okay. Has he been traveling?
So no medical history, no surgical, no prior surgeries, not allergic to anything, nothing's running in the family.
How old is he?
He's been in Missouri but hasn't traveled outside of there. He's 32, which is a little old for the heavy drinking, and maybe I'm being judgmental.
Are you kidding? Are you kidding?
Shouldn't he rein it in and get serious? He's still hanging out with his friends, drinking too much and doing crazy things on the weekends.
Might not drink too much compared to
Personally,
his
I think those genes should stop there.
Okay.
Right. So, do you have any hobbies?
He likes to hang out and do
He
all
doesn't-
kinds of
he
stuff.
doesn't fish, huh? He doesn't fish?
What his buddies do in Missouri? What did young kids do in Missouri? Whatever young kids do.
I have
It's
no
hot
idea.
in Missouri. What do you do in Missouri? It's
You
hot.
go to a river.
Okay, you hang out on the river. What do you do on the river?
Oh, you fish.
So he doesn't fish. What do the
Swim.
kids? What do the kids do? You're trying to fish, and these drunk kids are coming
They throw
by.
rocks and-
What do the drunk kids do? And they're floating down the rivers in their tubes.
Yeah, they're popping open beers. They're throwing the beer cans in the water. They're
So does he do
singing.
this?
He does that. Yeah. I mean, that's just what the kids
That's
do in
the
Missouri.
summertime. What do they do in the fall?
Then it gets a little too cold, and they just hang out, is what he says. No, he's not a hunter. He's not a hunter.
You're sure he's not a hunter?
I am sure he's not a hunter.
Okay.
What are you thinking, Kreutzfeldt Jakob?
No, not for this show.
Someone, a
Does he have any
politician.
pets?
He has no dogs, no cats, no birds, no animal exposure that you ask
Does he
him.
have any friends with pets?
No, none of his friends. Apparently they have too good a time to actually be responsible enough to care for another creature.
So no animal contact.
None that he reports.
All right.
Where does he live?
Missouri.
He lives in Missouri
No,
with
where
a
does he
with a
live?
bunch of his buddies in
Where
an
does he
apartment.
live? In an apartment. In a city?
It is. Yep, it's in the city.
There aren't too many of those in Missouri.
Yes, it's the one with that big arch by the
Oh,
river.
that one.
St. Louis?
You
St.
got
Louis,
it. You
St.
got
Louis.
it.
Is it St. Louis?
Yeah, that would
no,
be the
it's St.
one.
Louis.
Oh, there you go. Take me to St. Louis, Louis. Don't you
know
that song? Don't
there's
you
a
know that
virus
song?
named after that city.
It's an encephalitis virus.
Yeah.
It's a flavivirus. How did I know
Flavy.
that? Okay, Flavy. Flavy.
He's actually got a fever when you see him, 39.2.
Mm-hmm.
How many? Okay, can you tell me what the periodicity for this fever is?
You're getting it from this 30. He says periodically he'll have fever for a few days, and then he'll feel better for a few days, and then he'll come back.
Okay.
So like a week apart or something like this. And
Yeah,
it
maybe.
comes up to here and it goes back down again. And how long did you say?
He's been feeling crummy for 3 months now.
3 months. Okay, so I guess we have to start asking for tests now.
well, he
Now
doesn't-
we're going to give you a few tests, and then maybe you could ask a little more background. He's got this stuff going on, so he gets a chest X-ray,
Right, because
Okay.
he's got brown skin.
and he's not looking so good, right? He doesn't look so good. The chest X-ray shows fluid around the lungs, and he's got some increased, we say, opacification in both upper lungs. So he's got some fluid around the lungs, and it's little white if you look at.
There are other
And
things too when you're floating on rivers, by the way.
and he's got a bunch of eosinophils.
Roger.
30%
30.
eosinophilia.
Yeah, 30 is a big one.
Right?
I know.
You can't say it.
I'm not saying.
Now, what were you starting to say?
I'm
there?
I'm thinking, as Judge Benny used to say.
You said people do other things on the rivers. What is it? You think the rivers is key? You think that's important here?
I definitely think so.
But he's not really in the river, right? He's sitting on a tube. He's floating down the river. He's drinking a bunch of
Every
beer.
now and then you might get hungry.
Okay. So you think he might get hungry?
He might get
So
hungry.
should we ask him, like, when you're floating down the river, what are you eating?
What'd you have for lunch?
Okay. Well, they usually don't bring lunch
No.
with them. He doesn't
No,
bring
they
any
don't.
food with them.
No, they don't have to do that because near St. Louis, maybe not on the Mississippi, but certainly on the drainage system to the Mississippi in Missouri, they're all karst. You know what karst means? Okay. So these karst rivers have huge populations of fish
Okay.
and the food for the fish. And a lot of the food is in the form of hard-shelled organisms.
Oh,
So
And
should
one
we,
of those hard-shelled organisms happens to be a crayfish.
crayfish, right?
Right. So they have a lot of great smallmouth bass fishing in Missouri
Yeah,
as well
okay.
as trout fishing. So when you're floating down a river and you're getting hungry and you've got a few beers and you might decide to pull over to the side of the river and make a little fire and then go hunting for some of those crayfish.
Or maybe not cook them at
I mean,
all.
you might not, but I know that this is a common behavior because it's easy to do, and they're delicious, by the way.
Okay.
This is like...
Has he ever noticed any insect bites of any sort?
Is this...
lots
wait.
of yeah. I
Yeah.
mean, when he's
Lots
floating
of
down the river, right?
Of course,
he's
you're
not,
getting
they
bitten alive.
never. Yeah, they're getting bitten alive.
They get
But
tick bites?
You didn't answer this question for me, did
So, did you want to know? Did he eat any
I'd
crawfish while
love
he was
to
floating
know
down
if
the
he ever
river?
did.
So he did.
Roger that.
Yes,
I'm
and he
gonna
did not stop. And he did
asking
not
questions.
stop to cook them.
I'm stopping to ask questions.
I told you
He ate a
didn't cook
huge
'em.
one.
I am not.
He actually is
asking
quite
anymore.
proud of the
You know what?
size
I'm
of the
not
one.
asking anymore.
No, his friends made him eat it
How
on
long
a
ago
dare.
was that? How long ago
And
was
it was
that?
back in the summer.
Oh, but of course.
He ate, but he did not say it tasted good. He did it on
And
a
I'm
dare.
really
He had a
sorry
couple
that I'm
beers.
sitting here spoiling this for everybody else because
Well, no,
No, but you're not going to tell us
Dickson
what it
only-
is, right?
Dickson only thinks he spoiled it for us.
Because, okay, they'll be able to get this.
Yeah.
All right, this is excellent. I don't think we need more because it's easy. It's easy to find out.
Yeah,
what
let
this
people
one is.
start thinking about what this might be. So again, we got a 32-year-old male, likes to drink a bunch, likes to float down the rivers, likes to hang out with his buddies.
until next week.
All right, would you like to hear one more email?
Of course.
Would you like to read it?
Sure.
It's from our friend Robin.
Yeah, I did see that.
You have to talk into the mic, though, Dickson.
Well, let me find it on my computer
Oh, my gosh.
He's finding it on his computer and speaking into the nether, so we may not hear him.
It's because I think Daniel would enjoy it. Do you see the show
It's
notes,
going
by the way?
to take him a while to find this. Yes.
Right. So Robin writes, my 4 episodes of malaria were in childhood before my teens in East Pakistan, now Bangladesh, and West Pakistan, now Pakistan. That's the closest we come to cohabitating with a photosynthetic organism. The malaria parasite is descendant from a lineage of photosynthetic dinoflagellates and still carries in its genome a few vestiges of its ancient ancestral photosynthetic machinery. Aha! A far cry from corals and free-living non-photosynthetic microbes, which associate with photosynthesizers that act as symbionts, endosymbionts, and even become acquired organelles. Some New Guinea tribes had a 95% prevalence of asymptomatic malarial parasitemia in adults. The prevalence among adults in Gambia was 70+%. If in deep time past the parasite could have found a way to ride along through the vertebrate germ cell line, they might have found a way to earn their keep and become an endosymbiont.
That's funny. That's funny.
Old wives' tales, these are best spread by GYN specialists. I don't think so. Swollen leg, the patient usually does not worry if the condition is painless. Nighttime thick and thin blood smears from a finger stick, Guim sustained, could be examined. This is responding to the case that we just discussed.
Oh, sorry about that.
First thing to treat elephantiasis is, of course, penicillin or other related drugs if the patient is not allergic to them. Prompt attention to treatment for episodes of local cellulitis and special attention to local hygiene and cleanliness to prevent cellulitic episodes can be very helpful. Albendazole with ivermectin should deal with microfilariae, doxycycline against Wolbachia, get Wolbachia, it's an endosymbiont in Wuchereria, which causes the pathology, just like it does in Onchocerca volvulus, should permanently eliminate microfilaria and halt progressive elephantiasis. If the ulcer looks like leishmaniasis and shows no improvement with PCN, a PCR is in order, but since it is not reportable, neither the CDC nor State Health Department would be persuaded to pick up the tab. The physician may bill TWiP. Take what insurance pays. That's what TWiP stands for now. But the laboratory service is another matter. Meanwhile, fluconazole may be an option until laboratory results. Miltefosine may be a consideration then.
Miltefosine.
Miltefosine. Okay. Somebody want to read the next one?
All right. Yeah, why don't Daniel read it? This is good.
Yeah, absolutely.
Good way to engage Daniel.
Oscar writes?
Yes.
Right.
That was a sneaky way to get someone to listen to the back episodes. Well played, Dr. Griffin. Well played. Also, keep up the experimentation. You all have good instincts. Follow them. I'm wondering if he was referring to, we're trying to figure out why they call it parasitism instead of parasitology. I wonder if that cuts down on our ability to get found on iTunes. But anyway, the world needs a new car talk, weekly enlightening mysteries. You could do it. At least 3 different mysteries presented and solved per episode. There are some back episodes where Dickson and Vincent trade off questioning each other through the whole presentation, and it really works. Instead of the car talk guys questioning the listener, you question each other, and it steps the listener through the paper. Here are some free ideas instead of case or paper or presentation. I'm going to use the term mystery. All 3 of you should laugh at least 3 times per episode. I definitely made the quota this time. I was very entertained.
I'm afraid I did.
The repartee.
And all for the wrong reasons, too.
Instead of listeners call in, have a smarter person than us call out, perhaps to enlighten a mystery or present a mystery or share news or report on location. That parasitologist from Mayo, Bobby Pritt, that you had a couple of months back would make a great regular. Not too often, not too long. Like when some professor from MIT would call in to yell at Tom and Ray. Okay.
Correct.
Yeah, no, I really enjoyed that Bobby Pritt episode. And I was listening to a few lectures. She's got some interesting parasite cases.
Well, maybe we should call her for the diagnostic tests that we saw.
Yeah, that would be.
How do you run a knot test?
We could actually, when you do your case, we could call her and have her listen in, right?
Yeah, that would be fun.
We could,
but we don't want it. We don't want it to be guest, right?
No, but we could make her a regular addition to the show. This is the lab aspect.
Now, figure out what kind of mysteries the bit you're doing is. Figure out some sort of taxonomic classification. Get a few solid kinds so you and the listeners can start to catch a rhythm. 4, know how much of the mysteries you want to give away going in. And I think we've decided to give away less.
Right.
Have at least one mystery that goes on for more than a week. Maybe an easy and a hard one. Perhaps the smarter person than us could give all 3 of you a mystery that is outside your expertise and even outside the scope of parasitology. The Car Talk hosts weren't afraid to blindly grasp for an automotive engineer's solution to a relationship problem that had nothing to do with cars. You shouldn't either. It keeps you extra human. Mm-hmm. Here's a mystery presentation idea. 1 question. Give a really short background and have people email or perhaps fill out a form where they get to ask a question and take a guess. If they get it the first week, on the first question, they get lauded. I think this is like Name That Tune or something, right? If they ask a question and guess wrong, it goes to the next week with a new person where the short presentation, 1 or 2 sentences, and then a summary of all questions, and the new person gets to ask a question and guess. The longer it goes, the greater the prize, the better the prize. If all 3 of you took turns coming up with the mystery, you could compete with each other as well as to who could stump the listeners the longest. Oh, I like stump. Maybe you could call it Chump the Stumps for a shameless but not copyright infringing homage to Car Talk. 7, have fun. Eager listening, Oscar.
So what is Chump the Stumps based on?
That they were Stump the Chumps.
Stump the Chumps. That's what they were?
That's what they were.
Okay.
That's what they call it when they phoned in. So I think the parasite of choice here to come up with a much smarter approach to this would be Mycroft-Filaria.
Okay.
Get it?
No.
Mycroft-Filaria?
Yeah, I get it.
What was Sherlock Holmes' name for his smarter brother?
The one who hung out at the club and never
Mycroft.
came out? Mycroft.
Mycroft. Thank you. Mycroft.
Yeah.
It was a sort of
Yeah,
play on
Mycroft, right.
It's a play on words. Mycroft Filaria. Mycroft Filaria. So you get it?
Dickson.
Okay.
That's it for TWiP.
That was pretty deep.
84, Dickson. You'll find it at iTunes at microbeworld.org/TWiP. They're all there, and you can listen to them all. They're free. They will always be free. We will never charge for TWiP,
No.
TWiV, TWIM, or urban agriculture.
That's correct.
That is my promise.
And mine.
Dixon wants to charge $10 an episode
Stop it. It's a lie.
because he loves money.
This is a lie. Vincent's nose just grew 5 inches when he said that.
Dixon, where can people send email if they want to ask a question or propose a solution to the mystery? Can we call it a mystery? A parasite mystery?
You're going to tell him where to send it, aren't you, Vincent?
TWiP at twiv.tv.
Bingo.
And Dickson Despommier can be found at tricanella.org.
Yes.
It's relevant to parasitism.
And medicalecology.org, too, because that website is still up and running, and there's a lot of this information on there. If they look
.com.
for it.
It's a .com, isn't it?
I think it's a .com. Yeah, I think
Dixon, thank you.
You're welcome.
Always
is
good to
fun.
have you.
I've learned so much. It's true.
One cannot tell with you often.
No, I've learned a lot.
Daniel Griffin is here at Columbia University Medical Center. Thank you, Daniel.
Oh, it was a pleasure again.
You see, we get good emails. People like you.
I appreciate. We're actually all, people may not realize, but we're all actually fairly sensitive individuals. So we appreciate the emails. It helps.
That's right. We have
Every
fragile
little bit
egos.
helps. Yeah, we do all have fragile egos. So thank you for being sensitive to that.
Fragile X's. Fragile Y in this case, right? Because that's X is a female thing. Okay.
All right.
I'm Vincent Racaniello. You can find me at virology.ws. Music that you hear at the beginning and end of TWiP, which I happen to really like, is composed and performed by Ronald Jenkins. You can find more of his work at ronaldjenkins.com. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back very soon. Another TWiP is parasitic.